Good morning, everyone. My name is Dipto Kundu from the University of Rajshahi in Bangladesh. Today, I will be presenting our metagenomic study on microbial shifts in upper gastrointestinal carcinoma. Let me start by walking through our methodology. We sourced 380 raw samples, 16S rRNA sequencing samples from ENA Browser representing patients with upper gastrointestinal carcinoma. After rigorous quality filtering and applying Fred score, chimera removal, and length filtering, we retained 282, giving us 74% retention rate. These 282 samples yielded 2461 ASVs, that is amplicon sequence variants, and our bioinformatic pipeline was built on QIM2 using data2 for denoising the pair and first Q reads. Primers and were trimmed using CUDA-depth, sequence were denoised, merged, and truncated through data2 error learning model because your data2 is self-learning model. For analysis, we performed alpha and beta diversity assessment, including Shannon, Chawan, Simpson, and in beta diversity we use Bray-Curtis, UniFrac, and for the significance we test the Parmonova and identify differential taxa, and finally the differential pathway or pathway enrichment pathway in this differential bacteria, and we predicted the metacyclic pathway using LDA score greater than 2.7 or 2.8. Here are the results of alpha diversity. We first finds that the microbial richness or evenness within the individual samples. We compared four sample types, that is mainly we divided the samples in two category for based on the sampling method. uh, the samples from carcinoma tissue biopsies, the samples from normal tissue biopsies is the one group, and the samples from carcinoma mucosal swab and normal mucosal swab. Strikingly, carcinoma tissue biopsies showed significantly higher species richness across all matrices, Shannon index, Chow1, and observed ASVs compared to normal biopsies. Carcinoma mucosal swab also showed increased evenness related. We show the slides. We can see the differences and the richness and evenness between the normal and carcinoma samples. And if we go to the beta diversity, the tissue biopsies showed strong compositional divergence between carcinoma and normal tissues, and mucosal swabs showed a more moderate separation between group, but still meaningful. After that, we need to know which specific microbes are driving these differences. We find in mucosal swab there are 32 significant differential ASVs, that differential bacteria species. 12 are enriched in carcinoma and 20 in normal tissue. Among those enriched in carcinoma, Helicobacter pylori stood out with a log-fold change of 2.05, alongside Acinetobacter sp, one species of Acinetobacter, one species of Staphyia, and several pivotal species. Normal swabs are dominated by Novospongobium capsulatum and Nitrosporidium sp, Rohita mucilinase. In tissue biopsy, we identified 51 significant bacteria, and 12 enriched in carcinoma tissue and 39 in normal tissue. If we go to the cross validation, we see that the Helicobacter pylori and Acinetobacter, one species of Acinetobacter, arise in both uh, sampling method, that is its consistent enrichment across both biopsies and swab validates our approach that we gives it gives us confidence in finding, because Helicobacter pylori is one of the most established pylori in different microbial paper that is related to UGI carcinoma. But our concern is with these bacteria, that is one is the Istella hyphensis. This bacteria is not well established, and this has the high LDA score. It's greater than 3, and it is the mostly impactful bacteria that we can see, and it is the gram positive bacteria which can be uh, make the suitable or make the environment for the carcinogenesis. And also the streptococcus rohit novospongium are the as usual bacteria that's arrived in our normal tissue, but the bacteria with the genes of these bacteria, of the significant bacteria follows, and our concern is to find out the drug and therapeutics before the carcinogenesis. Carcinoma swab and 8 enriched in normal swab. My work is I continuing this work. I didn't complete my research fully. I next I will find the key bacterial specific genes in this bacteria are driving these abnormal metabolic pathways seen in cancer patients, and then we go to the molecular docking for finding drugs and therapeutic agents, a step toward a new cancer treatment. Thank you for your time.